Now this is the second part of the molecular assembly. So from last time we've already talked about the molecular assembly in carbohydrates and the proteins. Now we are going to look at other biomolecules. Vesicle. Sorry. Structure is the molecules, right? The starting molecules. The vesicle, you have two tails with one very high polar head, right? Very high polar head. But the surfactant, you have only one tail, and also with a very high high polarity head, carboxylate ion. So the difference is the size of the head and tails. So size of head and size of tails are very important in order to arrange themselves or assemble themselves together into a supramolecular structure. So actually, uh, there are functions calculated from the volume of the tails and the volume of the head. So we call this as the critical packing parameter. So if the head is so big and the tail is so small, so as you can see, the packing parameter over here is the volume of the tail divided by the head roughly. So if the tail is so small and the head is so big, so what you can see is head trying to stay away from each other, as you can see at the carboxylate group, trying to repel each other by electrostatic repulsion. But the tails are London force, right? So at the end we have spherical micelles. But if the tail is getting bigger, right? So it transformed from the spherical micelles to cylindrical micelles, because they can then somehow packing themselves into cylindrical micelles because the head is getting smaller or maybe the tail is getting bigger. And for lipid bilayers on vesicle and cell membrane, it is somehow the critical packing parameter is around 0.5 to 1. So they can have the lipid bilayer kind of structure here. And if it is cylindrical, meaning that the size of head and tails are quite the same, they have a lamellar faces, right? And if the tails is very huge, but the head is very small, then we can have reverse micelles or microemulsions over here as a result. So size of head and size of tail, or the stereohedrons, is a very important thing, plays a major role in the self-assembled properties of this kind of amphiphilic molecule. Now, from our knowledge about the fatty acids, which is the major part or major part in the major component in phospholipid, sphingolipid, and also soap molecules. Um, the fatty acid can be divided into two major types, the saturated and unsaturated. And everybody have to know that the saturated fatty acid normally have higher melting point because of the well-organized structure by and by the packing system of the molecules. But the unsaturated fatty acid with the double bond cis isomer, this prevents them from packing close together, so it's quite fluid. It is exhibit lower melting point, right? So normally unsaturated fatty acid uh, has lower melting point comparing with the saturated fatty acid. So the fatty acid composed um, condensed with a glycerol, thing like that, making phospholipids and sphingolipids. And those phospholipid and sphingolipid is a major component in the cell membrane, in the membrane of the Golgi apparatus, in the cell membranes of the nerves here in my picture. So comes to the assignment, come to the question. The question is, which part of the structure over here contain more saturated fatty acids, right? So Golgi body, cell membranes, and the nerve cells for body part and the axon part, all of these constructed from the lipid bilayers. And the lipid bilayers almost always composed of sphingolipid and phospholipid. So the fatty acids that is um, that is composed there in the structure of phospholipid and sphingolipid can exhibit some properties. So what I'm going to ask is comparing between Golgi body, structure of membrane of the Golgi body, and the cell membrane. Which kind of membranes contains more ratio of saturated fatty acids? And on the right hand side, it is the nerve cells. The nerve cells has two parts, the body part and the axon part. The question is the same. Comparing between body part and axon part, which part contains more saturated fatty acids in the cell membrane? Okay, so this is the questions. I hope you can find the answer very easy. See you next time. Thank you very much indeed.